My name is Thomas Holm Petersen and I'm corresponding author on a paper that will publish today in Science Translational Medicine. The work comes from an international and cross-sector collaboration between NMD Pharma, Aarhus University, Leiden University Medical Center, Center for Human Drug Research, Ohio State University, and Next Generation Precision Health at University of Missouri. The paper reports on using skeletal muscle-specific CLC1 chloride ion channels as a novel drug target for restoring muscle function in myasthenia gravis. Skeletal muscle contractions are the underlying power generator for movement, and well-functioning muscle is therefore essential for human behavior. Activation of skeletal muscle contractions involves successful transmission of action potentials from motor neurons to muscle fibers. Such transmission is initiated when a neuronal action potential arrives at the motor nerve terminal, where it triggers the release of acetylcholine into the synaptic cleft. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors on the muscle fiber surface, causing these receptors to open. Activated acetylcholine receptors conduct an inward-directed excitatory membrane current that depolarizes the muscle fibers locally at the neuromuscular junction. In addition to this excitatory current, there is, however, also a powerful inhibitory current that flows through CLC1 chloride ion channels. These channels are expressed at the neuromuscular junction, in the periphery of the junction, and also along the sarcolemma of the muscle fiber membranes. Inhibitory CLC1 current effectively shunts or short-circuits muscle fiber excitation, and neuromuscular transmission is therefore highly dependent on the size of this excitatory current versus the size of the inhibitory CLC1 current. In healthy muscle, excitatory current outweighs inhibitory current, and neuronal action potentials are therefore reliably transmitted to muscle fiber action potentials, but this can drastically change with disease. Myasthenia gravis is caused by an autoimmune response targeting membrane proteins at the neuromuscular junction. This leads to loss of functional acetylcholine receptors at the junction and morphological changes to the junction. In turn, this causes the excitatory current during neuromuscular transmission to be reduced. Under these conditions, the CLC1 inhibitory current becomes too dominating, and excitation of muscle fiber action potentials and muscle fiber contractions fail, resulting in the muscle weakness and muscle fatigue that cause the symptomatology of myasthenia gravis. In our paper, we demonstrate the pharmacological inhibition of CLC1 can recover neuromuscular transmission and restore muscle function in myasthenia gravis. We present a small molecule inhibitor of CLC1, NMD670, and demonstrate that this molecule can enhance neuromuscular transmission, restore muscle function, survival, and performance in rats with experimental autoimmune myasthenia gravis. Importantly, we also qualified NMD670 for clinical trial and demonstrate that a single administration of NMD670 caused statistically significant and clinically meaningful improvement in muscle functions in patients with myasthenia gravis. We consider this to be the first clinical proof of mechanism for CLC1 inhibition as a differentiated and muscle-specific treatment approach to restore muscle function in myasthenia gravis and potentially other neuromuscular diseases. Thank you for your attention.